Hi, everyone, and welcome. My name is Kimia Grigoriev. I'm the founder and president of the Harvard Extension School Psychology Student Club. We're so grateful to each and every one of you for joining us today. A special thank you to our brilliant speaker, Dr. Michael Levin, outstanding scientist and educator, Vannevar Bush Professor of Biology at Tufts University, and Director of Allen Discovery Center. And also very likely, and I will be shocked if this doesn't happen, most likely a future Nobel Prize winner because of his incredible work in science. I will provide the links again in the chat in ways that you can follow his work. We are super honored to have him and so grateful because I know he's incredibly busy, but he's making time to be here today and sharing something incredible with us. I'd also like to thank Dr. Max Krasnow, our wonderful advisor, who always goes above and beyond for his students. Thank you for your ongoing support and guidance through the years. I also like to thank our leadership team. Thank you guys for always helping me with my shenanigans. For our live participants, a quick reminder, this event is being recorded. If you don't wish to be in the recording, to protect your privacy, please ensure your name and your photo are hidden and your camera is off if you prefer. If you have any questions for Dr. Levin, you can submit them in two ways. Use the Q&A section with your name, or you can submit them anonymously if you prefer. Depending on time and our speaker's preference, we might be able to take questions by raising hands. While we may not be able to get to every question, because I know time might be limited, we'll do our best to get every question answered. I will also provide ways you can connect with us in the chat, our social media, our website, and our Spotify. And I don't want to take too much time because I know Dr. Levin has incredible things to share with us, but thank you all for being here. And thank you so much, Dr. Levin, and welcome.
So much, and thank you to Kimmy and Max and everyone else for having me here. It's a real pleasure to speak to you. I will absolutely answer everybody's question. If we don't get to it today in the slot that we have, Kimmy has my email address, so please contact me, and I will answer all questions. What I want to talk about today, and this is a new talk, one that I haven't given before, so let's see how it goes. What I'm going to talk about today are the challenges and the progress that is being made in being able to recognize novel kinds of minds. If you're interested in psychology, one of the key questions is what are all the things that are part of that science? In other words, what are the things that have psyches for us to analyze? And what I'm going to argue is that I think inevitably, as evolved beings with very specific cognitive and sensory architectures, we find it difficult to see certain kinds of intelligence, and we need to work on that. So there's a particular Buddhist vow that I think is really interesting, which says that sentient beings are innumerable, and yet we're supposed to liberate them all. And so, in order to liberate them, we have to understand what it means to detect some of these innumerable beings that are all around us. And this is a sculpture that I really like that shows that when you look at it in one sort of perspective, it's a bunch of silverware, it's forks and knives and spoons and things like this. But when you shine some light through it, you find out that there's a deep structure underneath that is not at all obvious. And so a main theme of this talk is going to be that perspective is key, and that looking at a system and trying to understand whether it has a mind, and if so, what kind it has, is basically an IQ test for the observer as well. That is for us. It's really easy to miss these things. So I want to start out by making an analogy to the electromagnetic spectrum. So in the days before we had a good theory of electromagnetism, there was a wide range of phenomena that were all thought to be different things. So lightning, static electricity, magnets, light, all of these things were thought to be different. And what the good theory of electromagnetism allowed us to do is two things. First of all, to understand that they are all actually fundamentally one thing. They are all on the same spectrum, and there's a parameter that will shift you between the properties of gamma rays and X-rays and visible light and whatever, but they are all fundamentally one thing. So it's a major unification. Okay, and the other thing that it tells us is that while our native senses only allow us to see this tiny bit of that spectrum, so that visible spectrum that you can see here, this is a log scale, you can see how little of it we're actually sensitive to. while we are only natively sensitive to that log scale, with the proper theory and the proper instrumentation, we become sensitive to a much larger universe of information that's out there, and be able to actually make useful devices that that improve the quality of life for everyone. So I think this is the kind of thing that I would like to do for the topic of cognition, and is to do a little bit of unification and show that different things that seem different to us are really part of the same cognitive spectrum, and that by having the right theory and the right instrumentation, which the theory enables, we will be able to recognize things that to which we are now basically blind. And so of course, I'm not the first to try this. Rosenluth, Wiener, and Bigelow were already in 1943 talking about this kind of scale from passive matter up through complex metacognitive minds. And so this idea has been out there, but what I've been working on are frameworks to help us recognize, create, and ethically relate to very diverse different kinds of intelligences. So familiar things like brainy animals, but then also colonial organisms and swarms and synthetic life forms, and AI, whether purely software or robotic, maybe someday alien beings, and something else that I'll talk about at the end of this talk, which is even weirder than any of those things, which are patterns within media. And my goal is to do this in a way that moves experimental work forward. So in other words, not just philosophical ideas, but something that can help us make new discoveries in biomedicine and synthetic bioengineering, and actually will lead us to some more sophisticated ethics, which I think is really important. So I've shown this many times before. This is my spectrum of persuadability, which is basically this idea that when you have different kinds of systems, whether they be engineered, evolved, or some combination thereof, what you're really saying when you make cognitive claims is that you're making a hypothesis about what set of tools are appropriate for in fact optimal for interacting with that system. It might be physical rewiring, it might be the tools of control theory, or cybernetics, or it might be behavior science, or it might be psychoanalysis or friendship, love, things like that. And the idea is that you can't just sort of decide where things are along that spectrum. You have to do experiments, because we don't know. We are not good at guessing. And so another kind of analogy we can think about, and I think this is kind of apt, and it takes us back to this Buddhist idea of innumerable beings, and what does it mean to be innumerable. If you think about how numbers were discovered throughout the history of mathematics. We had some counting numbers, and then eventually people figured out zero, and then we had integers, including negative integers, and then the rational fractions, and then algebraics, and then very weird transcendentals like this. And what happened each time that a new category of number was discovered is that it needed a conceptual leap. It needed a way to think differently, because now you're coming out of the set of things that you're comfortable with into a new higher level of thinking. So you have to make a conceptual leap. All of these leaps broke conventional boundaries. So the things that you thought were limits of your concept turns out to be not a limit, and you can go beyond it. And it actually caused quite a bit of disruption in society and things like that. And so basically, what I want to do today, and I'm going to hopefully get through to this. I'm not going to have time to talk about this yet, and that's still very much being developed. But what I want us to think about are all the different continuously enlarging sets of things that we can think of as cognitive beings, and each one of these is, and especially as we get up here, we're really starting to look at various infinities, and in particular different degrees of infinities, because these are not all innumerable in the same way. So we'll go down the line, and I'll show you what I mean. So what we want to do here is get our conceptual frameworks and hopefully useful applications in line, so that we are not disturbed when we find these things. Like this poor guy who drowned at sea when discovering kinds of numbers. That he drowned himself at sea when discovering kinds of numbers that were thought to be impossible. And so what I'm going to show you is no doubt disturbing in certain ways, and it makes things for sure more complicated for us scientists who like to come up with simple categories of things. But I do think it makes both life and science somewhat more beautiful. So this is kind of a standard picture, at least in the Western world, of what this is a classic piece called Adam Names the Animals in the Garden of Eden. And so the idea here is very much rooted in anthropocentrism, so or at best brain chauvinism. But basically, what you have here is Adam. He is different than all of these other creatures. It's his job to name them all, meaning to identify their true nature. And we know, or at least we think we know, that there are discrete categories, right? So each of these things is kind of a discrete category. And so what I'm going to argue is that this picture has to go, and it's going to be replaced by a very different picture. But in case you think that this is just kind of a problem of the Western world, it really isn't. I've had these discussions with many people in the Buddhist, Indic, and other traditions, and there are aspects of where the field of diverse intelligence is going that are just as disruptive in those traditions. So I think this is a kind of a fundamental shift that's happening. So the first thing just to remind us is that intelligence in brainy organisms is not hard to see. We can, so here's this little flying squirrel. What's he doing? Well, he's setting up a murder scene, right? So there was an accident. It's very clear. He knows exactly what's going on. Sometimes, up there, just put it right on the neck, and then he's gonna actually look up and see are our mom and dad watching. To make sure that this is, are they looking? So the idea is, right, he has a very good theory of mind. He understands what their reaction is going to be. He knows that they want to take care of them, and we can tell, we can see immediately in these kinds of brainy mammals that they have minds. It's obvious because they're operating on roughly the same spatio-temporal scale as we are. Right, it doesn't take decades or hundreds of years to watch this animal think, nor does it happen in milliseconds. They operate in the same space in which we operate. They have similar goals to us. Okay, so we can see. Although I will point out that even with brains, things are actually not very simple. Karina Kaufman and I did a review recently of these kinds of really weird cases where you see individuals with drastically reduced brain hardware and yet normal or above normal intelligence. And there are many cases of remarkable high performance, whether that be in savant syndrome or just people who don't know until very late in life that they're actually missing huge chunks of their brain. So it's quite actually not clear what is the relationship between brain real estate and cognitive performance. So even in those cases, another thing that's true for conventional beings like us is that when you look at an embryo and you see, I don't know, hundreds of thousands of cells at the beginning, the reason you say there's one embryo is because we know that under normal circumstances, all of these cells are committed to the same journey in anatomical space. We know what they're going to build. They're going to build one being, and that's going to be our one embryo. But in fact, if you make little scratches in this blastoderm, the original work was done in ducks, and you make these little scratches. Each one of these regions will self-organize its own embryo, and eventually you get multiple twins and triplets and so on. And so the question of how many cognitive beings are actually in, quote unquote, this excitable medium is unclear. It's not set by the genetics. It's not determined. It could be anywhere from 0 to probably half a dozen or more individuals, full-on cognitive individuals that can emerge from a being like this. And of course, in adult patients, this story also has analogs in terms of split brain patients and dissociative disorders, in terms of how many actual minds are within any kind of a physical substrate. So, but it gets weirder than that, because as we know now, there is intelligence outside the brain, and we need to make a conceptual leap. We need to understand that intelligence is way older than the development of brains, what we're going to talk about here is a model system that we use, which are cells as a collective intelligence operating in anatomical space. So this is what we are all made of. This is called the lacrimaria. It's a single cell. This is a free living organism, but all cells have amazing capacities to solve all kinds of problems in their own domain. So we are made of an agential material that has preferences and goals and agendas and so on. In fact, even within our cells, just the molecular networks themselves are also not just clockwork mechanisms. They're actually capable of 6 different kinds of learning, including Pavlovian conditioning. And this is, you don't need the brain, you don't need cells, even just the molecular networks by the virtue of the mathematics that controls their behavior, they are already capable of different kinds of learning. And we are currently building devices that will let us to train these things for applications like drug conditioning and so on. And so what we are assembled in a kind of multi-scale competency architecture where there is learning, memory, goal-directedness, problem solving at every level of organization and in all kinds of weird spaces. And I'm going to use the word intelligence many times. When I say intelligence, I mean William James's definition, which is some degree of the ability to reach the same goal by different means, and the degree of whatever ingenuity it has to reach those goals while it's being blocked or derailed or prevented from those goals. That's the degree of intelligence that it has. And so, as I said before, we're pretty good at noticing intelligence in the three-dimensional world, things like this. But biology actually doesn't take that 3D space any more seriously than various other spaces that are pretty hard for us to imagine. We're pretty fixated on on movement in three-dimensional space. People in engineering and AI will often talk about AIs not being embodied because they don't have, well, at least many of them don't have a body with which to interact with the 3D world. But for biology, that 3D world is not the only place where they inhabit. And so the living systems do this prediction control kind of loop. So action, decision making, all of that, they do that behavior loop in many other spaces. The space of gene expression, okay, so there's a very high dimensional space of different possible gene expressions, the space of possible physiological states, and what we're going to talk about today, which is anatomical morphospace. That's the space of all possible geometric configurations of the body, which is the space that we all navigated when we got from being a single cell to being the organism that we are today. So we know that this journey is reliable. So we all get from a single cell to a human target morphology. But let's be clear, the reason I'm calling it this intelligence is not because it's reliable, nor because there's a rise in complexity. Those things are easy to get, and those are not intelligence. The reason I'm calling it intelligence is because actually this process has some fascinating problem-solving capacities, meaning that it has a navigation of anatomical morphospace that is has lots of commonalities with things that we see intelligent beings doing in three-dimensional space. So first of all, if we, for example, deviated from its goal by forcing it to start in the wrong position, basically we take an embryo, we cut it into pieces. Each piece does not make half a body. What they make are perfectly normal monozygotic twins, triplets, and so on. And they do it because even from different starting positions, they can still get to the correct region of morphospace corresponding to a normal human anatomy. Okay, so they can do this for starting from different positions. Some animals can do this during adulthood. So here's this axolotl. If you amputate the limb anywhere along this axis, the cells realize immediately that they've been deviated from the correct location of the morphospace. They will work really hard to get back there, and then they stop. And that's the most important thing about regeneration, is that it knows when to stop. When does it stop? It stops when the cells have reached the correct position in anatomical space. I don't mean the individual cells have reached the correct three-dimensional position. I mean the whole limb has reached the correct configuration in the space of all possible ways that cells could arrange themselves. This is one of my favorite examples of creative problem solving in that space. So this is a cross section from a kidney tubule, and you can see there's about 8 to 10 cells that normally work together to make this structure. Well, one thing you can do is you can create these polyploid newts that have many more copies of their genetic material in their cells. And when you do that, the cells compensate by getting bigger, but the newt stays the same size. Well, how can that be? How can you build the same newt if every cell is bigger? Well, that's because when the cells do get bigger, they also adjust themselves so that there's fewer of them to make the same structure. Now there's only 4 or 5 cells that are making the structure. When you make them truly gigantic, so here, this is a 5N or a 6N newt, the cells will bend around themselves and still leave the same hole in the middle. This is a completely different mechanism. This is cell-to-cell communication. This is cytoskeletal bending. So, this ability to use the tools that you have, your genetically encoded affordances, such as cell-to-cell communication pathways, cytoskeletal bending, and so on. The ability to use what you have in new ways to get your goals met is a standard thing we measure on IQ tests. That's, here are some objects, figure out how to use them to solve some problem you haven't seen before. So think about what it means to commit to being a newt in this world. When you're born, not only do you not know what's going to happen in your environment, but you also can't even count on your own parts. All of this is up for grabs. You don't know how many copies of your genetic material you're going to have. You don't know how big your cells are going to be. You don't know how many cells you're going to have. You're going to have to assemble that newt using the tools you have as best as you can, navigate that space as best as you can with whatever you have. And so that requirement of creative problem solving from scratch every single time, because everything is unreliable. Your genome is unreliable because it gets mutated. Your proteins, all of the lipids, everything in your body is unreliable. It is different than the way we make computational hardware today with error correction codes and abstraction layers that mean that when you're programming in a high level language, you don't need to worry about the copper and the silicon and things like that. The data stay where you put it. That is not the case in biology. And I think that this requirement to build these things with an unreliable substrate gives rise to the intelligence ratchet that we see all the way from the beginnings of life, whereby systems get progressively more and more intelligent because they are they're evolving in this kind of unreliable material where every step of competency hides even more information from selection, and thus your only hope is to ratchet up the algorithm and make it better and better. And so I think that's where the intelligence race comes from. So there are lots of other scenarios such as this, where we see these tadpoles that need to become frogs. When they rearrange their face, they don't need to start in the correct orientation. If we make these Picasso tadpoles where everything is in the wrong place, the eyes on the back of the head, the mouth is off to the side, they'll still make normal frogs. And that's because genetics doesn't specify some kind of system of rearrangements. It specifies an error minimization scheme. It specifies a system that is able to reach the same goal from different starting positions, and it can take corrective action. Okay. So now, how could this possibly work? Well, one of the things that, remember what we're doing here is we're exploring the collective intelligence of cells navigating anatomical morphospace as a model system. We don't have real aliens yet, but this is a very different kind of intelligence that operates on a different space. And what I'm showing you here are ways to that we've detected and begun to communicate with that intelligence as a kind of a case study for working with beings that are quite different. So the conventional intelligence that's in our brains, what it does is it uses neural hardware, so these ion channels and then electrical synapses. And what it does is process information to control muscles to move you through three-dimensional space. That was the sort of original functionality of nerve and muscle when it evolved, was to process information and move you through three-dimensional space in some adaptive way, depending on what's going on in your environment. And for this reason, neuroscientists can have this project of neural decoding, where the idea is we're going to read the electrophysiology and we're going to use some kind of a decoding method to extract the cognitive states of that being, the memories, the goals, the preferences from the electrophysiology that's happening here. Well, it turns out that this kind of scheme of encoding large-scale information in an electrical network and using electricity as a kind of cognitive glue that binds together individual neurons into a larger scale being that has preferences and goals and memories that don't belong to any of the individual cells, and that's the cognitive glue function. Turns out that that's extremely ancient. So every cell in your body has the same machinery. So what your brain is doing is actually homologous, not merely similar to, but actually homologous to the intelligence of the rest of your body. And what this electrical network was doing since the time of basically bacterial biofilms was thinking about something quite different. Instead of three-dimensional space, it thinks about anatomical space. And so instead of issuing commands to your muscles, what the rest of your body electrical networks are doing, and that what they did in embryonic development, and what they're continuously doing to upkeep your body against aging and cancer and those kinds of things, is to issue commands to cells to reconfigure tissues and organs to keep you in the right region of anatomical morphospace. And failures of that process we see as cancer, as aging, as degenerative disease, and so on. And again, what we've been doing for quite some time is that same process of neural decoding, except applied to non-neural cells. So could we read the intent of morphogenetic processes from the bioelectrics, the way that we read, the way that we can predict this, at least a little bit, behaviors by looking at the electrical activity of brains. And we do this again to gain insight into what kind of cognition is happening in this network, because it lives in a different space, it has different goals, it has different preferences. Can we learn to understand it? Can we learn to communicate with it? Okay. And so in order to be able to communicate it, the first thing you have to do is to be able to read these electrical signals. So we're going to use this beautiful bioelectric interface to try to interact with this new type of intelligence. So we developed some tools using voltage sensitive fluorescent dyes to read all of the electrical states of the cell. So this is an early frog embryo. This is a bunch of frog cells in culture. You can see there's very, very dynamic electrophysiology happening, and we do lots of computation to try and understand where these patterns come from, what are the rules regarding their propagation, how do they do pattern completion during injury and regeneration, and how we can visualize memories as attractors in some sort of landscape, some sort of computational landscape. So we have the ability to read their states. So that's a start. And here I'm going to show you a couple of examples. So this here is the appearance of what we call the electric face, which is the set of bioelectric patterns that tell a tadpole what its face is supposed to look like. So this is a frame from that video. So you can see here's where the eye is going to go, the mouth, the placos. This is where we're literally reading the memories of this cellular collective to see what it's going to build as a face. The bioelectricity is not only the cognitive glue for the individual cells in an embryo, it also holds together groups of embryos. So you can see here some amazing phenomena where if I poke this one in the middle, these guys out here find out about it very quickly because there's this bioelectrical wave that happens, and you can see that here, right? So they're communicating in a group. So electricity is really good at this, is pulling together competent subunits into a larger being, and here you can see. Together, there are some there's some neurons here that are spiking, but you also see these non-neural cells having their own bioelectrical life. So we can observe all these patterns, and it's fodder for all kinds of computational processes. But of course, the other thing we want to do is to send signals to it. We don't just want to read out what it says. We want to be able to talk to it. And how do we do that? No applied fields, no electrodes, no magnets, no frequencies, no electromagnetic fields, nothing like that. What we do is we hijack the normal communication interface that cells are using to talk to each other, which are these ion channels and gap junctions. So we use all the tools of modern neuroscience. So that means pharmacological agents to open and close channels. It means optogenetics to turn these things on and off at will in spatial patterns. And we can control the channels and the topology of the network with gap junctions. So I've shown you that morphogenesis is able to make decisions and solve various kinds of problems. I've shown you that the cells are kept aligned towards specific outcomes by virtue of electrical communication within a network as cognitive glue. And I'm now saying that we now have the technologies to actually insert messages into that medium. Into the bioelectricity is the cognitive medium of the collective intelligence of cells, and morphogenesis is its behavior. Okay, that's the analogy that I'm going for here. And so now it's time to say, okay, so what can we actually communicate to these systems? And remember what they're doing. They're solving problems in anatomical space. So whatever we communicate, we are going to read out what is it, how does their behavior change in anatomical space, the way that you would communicate with any other creature in 3D space and ask how its behavior changes. So the first thing that we can do is introduce a voltage pattern that says to them, build an eye. And we can introduce that pattern anywhere, for example, putting it on the gut here, and the cells hear that message, and sure enough, they build an eye. And so it's a very simple bioelectrical pattern. We do not tell them how to build an eye. We are not micromanaging it any more than I'm micromanaging the synapses in your brain. When I'm talking to you now, I give you a very thin communication conduit, which is our language. And I assume that you will take care of moving around your synaptic proteins in order to form memories and of whatever I say, right? This is the beauty of communicating with a cognitive system, is that you don't have to micromanage its states. It will do a lot of self-managing. You just have to provide signals on a fairly thin interface. So we can ask it to make an eye. If we only target a few cells here, the blue ones are the ones we target, they will take care of telling their neighbors that they need to participate, that there's not enough of them to form a proper eye. And so all of this tissue up here is not one that we injected. We only got these few cells. They get everybody else. So there's a secondary instruction here, much like ants recruit their friends to carry large objects and so on. And all of this works, okay, only because the only reason we're able to make it, we don't know how to micromanage the creation of an eye. It's a very complex organ. All of this works because the material is competent, because we can control what it does with prompts, not with turning individual genes on and off, not by worrying about which stem cells are doing what, by finding prompts that are meaningful to the system. Right. That is the another signature of having a cognitive system, is that there are arbitrary signals, such as this voltage pattern, which doesn't actually look anything like an eye. These arbitrary symbols that are interpreted by the system to exert and convinces it to do all kinds of complex downstream behaviors. That is the kind of thing you can do with this agential substrate of life. Another thing that's really interesting is that all cognitive agents maintain a boundary between them and the outside world, and that boundary is determined by the size of the goals that they can represent. So individual cells have a tiny little cognitive light cone. It's got a little bit of memory. It's got a little bit of anticipation forward, and it really only cares about things that happen at its scale. So the hunger level, pH, tiny little goals, tiny little homeostatic set points. But when cells join into electrical networks, suddenly they have the computational capacity to have a much bigger memory and anticipatory potential, and they can work on grandiose goals like building and rebuilding this limb. Individual cells have no idea what a limb is or how many fingers you're supposed to have, but the collective absolutely does, because it has that computational capacity to enlarge its cognitive light cone to these very large kind of goals in anatomical space. So one thing we can do with cognitive beings is to try to shift the border between them and the outside world. In other words, their own definition of self. We can try to shift it. And why would you want to do such a thing? Well, because a breakdown or a failure mode of the collective intelligence of the body is cancer. When individual cells disconnect from the electrical network, they just basically become like an amoeba in the rest of the body. To them is just external environment. They will go where they want. They do what they want. They're now unplugged from the goals of the larger collective. They're not more selfish. They just have smaller selves. And so one thing we might want to do as a cancer therapeutic, and this is what we've been pursuing, is the ability to reinflate the boundary between them and the outside world, and not kill them, not fix the genetic defects that they might have, but actually just to reconnect them to the rest of the network so that they rejoin a kind of mind meld that forces them to keep working on nice skin, muscle, these large goals instead of little tiny ancient unicellular goals. And so you can see that here. This is a nasty human oncogene can be expressed here. We've done the KRAS mutations, things like that. P53 and so on. And so there's the oncoprotein. It's right there. You can see it, it's blazingly strong. We haven't removed it, but it's the same animal, but there's no tumor. And that's because we force the cells to become, despite this, we force the cells to become electrically connected, and it's the physiology that drives outcomes, not the genetics. Again, a kind of a hallmark of cognitive systems, that they can learn and do things that are different from what their default genetics indicate. So just to wrap up this kind of this middle portion here, what I want to briefly talk about is the benefits of this. So I've told you some very strange things. I've told you that the processes of development, regeneration, cancer suppression that normally you would study in cell biology, developmental biology class, as the results of the rules of chemistry and complex chemical systems, sort of cranking away, following the laws of physics and chemistry. What I'm actually telling you is that view is very limiting, and that we gain a lot of capabilities and a lot of new discoveries that are practically useful by being open to the possibility that actually what you're actually looking at is a cognitive system with behavior in a novel space. And by taking that seriously, it allows you to use specific tools from cognitive neuroscience. And I haven't shown you all the things that we've done with SSRIs and optogenetics and all, active inference, all these other things that come from behavior and brain sciences that allow us to discover things that are very useful and that have not been found before. And in particular, where it's going to be useful is in the future of regenerative medicine, where basically all of the approaches to date are all here. There are all these kind of bottom-up things like surgery and stem cell transplants and CRISPR and synthetic biology and all of that. There's a whole area here that's just beginning to open up now of top-down controls having to do with training cells and tissues, having to do with agential implants that will go in your body and fix things because they themselves are living organisms. And all kinds of triggers, as I've shown you, and we could talk about many more things that we've done in regeneration of limbs and repair of birth defects. Again, not by micromanaging the hardware, but by providing signals that are convincing, that literally are convincing to the cells as a model of what it is that they should build, and they sort of drop whatever they were doing before and take up a new kind of construction that allows us to rebuild these things. So my claim is that the future, and not that far off. I mean, these things are already starting to come online. The future of regenerative medicine is going to look a lot more like kind of somatic psychiatry than it's going to look like chemistry. And that's because we are taking advantage of the goals, the preferences, and the intelligence of the tissue that we're dealing with. And so that is the practical benefit of applying some of these ideas of being willing to see mind in other substrates. So the next thing I want to go beyond. So now what I've told you, okay, so it's obvious brainy things are on the list, fine. Then I told you that, well, actually, even a bunch of cells that are not themselves brains are on the list as well. But you might think that's not really that crazy. I mean, they have the same materials, they have the same evolutionary origin. Okay, so non-brainy tissues can also make decisions and have memories and so on. So let's move forward from that. And now try to break down this standard kind of distinction between quote unquote life and machines. And the thing about that is to realize that for example, humans and their sort of unique agential glow that features in all kinds of stories of philosophy of mind and things like that. We're actually not only at the center of a continuum in terms of evolutionary and developmental history, meaning we all came from a single cell here. And if you think there's something special about humans, you have to have a story about where that appears. Does it develop gradually because there is no sharp dividing line? Well, there's another spectrum that we're at the bottom of, or I should say at the center of, which is that both technologically and biologically, numerous changes can be and are going to be made. And that's because, as I told you, the material of life is unreliable. The biggest thing that evolution emphasizes in living lineages is problem-solving capacity, the ability to deal with novelty. And that means that life is incredibly interoperable. So at all levels, from cells, tissues, organisms, whatever, we can introduce new engineered materials. They do fine. You will have some kind of a hybrid system. Life is very good at adapting to all of these kinds of things because it never really had any strong commitments to the contrary anyway. So it just kind of every time it does the best it can with what it has. And so all of these things are possible. This is I asked our graphic artist to draw me this to kind of ridicule this idea that people are working on like proof of humanity certificates and things like this. I mean, it's going to be completely impossible because we're not talking about machines that look like your toaster. What we're actually talking about are hybrids and cyborgs and things like that. We're trying to figure out if it's 51% of his brain has been replaced or not is a total fool's errand. So we need to start to understand what are we going to do for the status of these kinds of unusual organisms. And I want to show you a couple of examples. So here's a little proto-organism. He's swimming around. If I were to ask you what this was, you might say, well, this looks like some sort of primitive organism you got from a pond somewhere. And I would say, well, can you guess what the genome would look like? And you might think that it would look like the genome of one of these primitive organisms. You would be surprised to know that the genome here would be 100% Homo sapiens. So this is adult human. These are anthrobots made of adult human cells, donated tracheal epithelium. And we have a protocol that allows them to self-assemble into this kind of amazing self-motile little proto-organism. And there's nothing wrong with this genome. We have not changed any genes. We have not put any weird nanomaterial scaffolds. There's no genomic editing here. This is this is something that human cells can do when tasked with putting together some kind of a novel body, basically, that can live in this new environment. Now, these anthrobots, when you look at their gene expression, so this is a volcano plot of a transcriptome comparing gene expression of anthrobots to the tissue that they came from. About 9,000 different transcripts. In other words, half the genome is expressed differently in these guys, even though we haven't touched the DNA at all. We haven't put in any synthetic biology circuits. Their new lifestyle causes them to dip into their genome as a resource and radically change what it is that they're expressing. Not only do they have radically remodeled transcriptomes, they have some interesting behaviors that are novel. For example, they have 4 different behaviors that they can do. We can draw an ethogram with probability for the transition states between the different behaviors. And we found that if you place them in an arena where you had a bunch of human neurons and you took a scalpel and put a big scratch through it, so a big wound, what they will do is they will actually gather in this group, which is this Superbot cluster, and they will heal across the gap. So you lift them up 4 days later, you have this. Who would have thought that your tracheal cells that are sitting there quietly in your airway dealing with mucus and particles and whatever, if given the opportunity, could form a self-motile little organism that would run around and do things like heal parts of your body, right? It's an amazing thing. Nobody had any idea that this was possible. This is the first thing we tried. This isn't attempt 74 out of 5,000 things we tried. This is the first thing we tried. So I assume there's probably going to be an incredible number of novel and interesting and probably useful behaviors that these guys can do. Including, for example, agential interventions. So you can imagine putting anthrobots in the body, and you won't need immune suppression. They will do all these healing functions and so on. This is another novel proto-organism. These are called Xenobots, and they come from frog epithelial cells. And you can see each one of these circles is one cell. So this group of cells, it's this little cute little horse-like thing. It kind of moves around. You can see here it's interacting with these other cells. In a minute, you're going to see a flash, which is calcium signaling. No idea what they're saying to each other yet. But again, these cells are not manipulated in any way other than we liberate them away from the rest of the body. What does the rest of the body do? Well, it sends signals to basically bully these cells into being a boring two-dimensional skin covering for the animal. If you liberate them from those cells, they will actually do something quite different, which is to become these amazing Xenobots. So they have the ability to swim like this. Here you go. You can see them. You can see this thing chugging along. You can make weird shapes out of them. They have social behaviors such as this. Here it is traversing a maze. So you can see it's going to go here. It will take this corner without having to bump into the opposite wall. So it takes the corner, and then here it spontaneously turns around and decided to turn around and go back where it came from. Another amazing thing that they're doing is when you provide them with loose skin cells, here these are loose epithelial cells, they will work both together and individually to collect them into little balls. They will then polish these little spheres, and because they're dealing with an agential material themselves, these little balls mature and become the next generation of Xenobots. And so guess what they do? They run around and make the next generation and the next generation. So this is called kinematic self-replication. To our knowledge, no other creature on Earth replicates this way. They found this way of doing it because we've made it impossible for them to reproduce in the normal frog fashion. And there's something else that's really wild, which is that we were looking at the transcriptome of these Xenobots. Much like the anthrobots, there are hundreds and hundreds of genes that they express differently. One of the things we found is a cluster of upregulated genes compared to embryos. So embryos do not express this, but Xenobots do. And it is a cluster of genes related to sensory perception of sound. And so we thought that's wild. Why are they expressing genes related to the perception of sound? We thought, is it possible that they can hear? And so here's the experiment that we did. So we got our Xenobots. We're watching their movement. We've got a speaker underneath into which we can play various sounds. And you can see what's happening here is that here's compared to a Xenobot that's too young to move. It doesn't really do anything. So you see the sound is not passively moving it along. If the Xenobot isn't active, nothing happens. But here what you see is that it's got its normal curly kind of motion. You start to play the sound and it starts to do this completely different motion. You turn off the sound and it goes back to what it was doing before. So it reacts to, I don't know, let me see if this video, yeah, I need to do a zoom in. You can see this is the little guy here. And so it's doing this little curly thing. And then when the vibration comes on, it starts, you can see here, it starts swimming off into this different pattern. And then when the sound is going to go off, then it's going to stop again. So what you're seeing here is our attempts to take a novel being that has never been here before. So there's never been selection for being a good Xenobot. There's never been selection for being a good Anthrobot. These things are not in the standard evolutionary stream. And figuring out what do they do natively, what are their preferences. We're doing now learning experiments to see what they can learn and so on. But very importantly, we're trying to develop methods to learn to communicate with them. So if you want the Anthrobots to heal, what do you need to say to them, in what modality? If you want to change the way the Xenobots are moving, what kind of signal, right? And you can imagine doing all sorts of interesting things. Do they react to music? Can we give them specific stimuli to make them do specific things? But again, remember what this is. This is our attempt to try to communicate with an intelligence that is really not only not easy for us to understand, but actually not on the typical evolutionary tree with us at all. And so another way to think about this then is that the actual being is not the Xenobot itself. You can think about it differently, which is all of our collaborators, such as Josh Bongard and Sam Kriegman and Doug in my center who worked on all this. All of us, together with the AI that we use to simulate the Xenobots and try to infer some interventions and different ways to manipulate them, all of this together is a kind of organism, because all of this, the intelligence of all of these things, is being merged together to produce this kind of novel system. So we're starting to get now into really unconventional beings that have multiple biologicals, and then here's an AI that's also involved in designing these Xenobots. And so this is going to be more and more true because we, so this is ours, and then other people are going to do this too, making robot scientist platforms, which are simultaneously, there's going to be some kind of AI, there's going to be some sort of robotic platform that's going to build biological systems for, let's say, medical purposes and biobots and so on. And it's going to do this cycle of inference and design and hypothesis testing. And again, you can see all of this as a new kind of being that has intelligence baked in at different kinds, at different scales and different materials. All of this is a coherent thing that is solving various problems through its ability to communicate with the biology. And so, here's something that we put in a recent paper, talking about, like, how far can you push this idea that, okay, we know the conventional version, so here's the brain, here's the body, here's the environment, okay, nice and simple. Well, how can we sort of break down the boundaries for all these things? So you can imagine here's a radically different agent. What you have here is some kind of brain tissue in a vat. So this is a mammalian brain. Then there are some insect neurons. They're communicating with each other through an electrical interface. That whole thing is being watched by a computer, and the signals that they output control the behavior of a robot swarm in an arena. And whatever the robots are doing, there are humans out there with their little iPhones clicking on whether they like or dislike what the robots are building, and that gets fed back. And what it does is it rewards the various neurons with endorphins and other goodies. So you have this whole system, you have a whole cycle. All of this is present: brain components, body components, environment. But it's really hard to say which is which. Okay, they're all here, and they all make a cycle. But it's it could be distributed over the entire Earth's surface, uses many different other beings as components, the way that our bodies use cells. And so you can see that this basic scheme of brain, body, and environment can get really, really weird. And what's fundamental to this is not the scale or the materials or the problem space, but actually this loop, this ability to process information, take actions, and reap the consequences of your actions in some space. So I don't have, we're running out of time, so I'm not going to go into this in detail. But one thing I want to point out is, so far we've been talking about different kinds of biologicals, even okay. So the last thing was the hybrid systems, but still these are pretty complex. Anything having to do with a living cell is incredibly complex. And I want to point out, and anybody who's interested in this can take a look at this paper, the ability to have unexpected protocognitive properties, problem solving capacities, and other goals that they were not programmed for. Does not require life. It does not require cells. It doesn't require much complexity at all. Even something as stupid as a sorting algorithm. So things like bubble sort, these very simple deterministic 6 lines of code. People have studied them for decades. If you look at them the right way, you find out that they already have things like delayed gratification. They do these weird side quests that are separate from the sorting of the numbers that the algorithm is telling them to do. And so this idea that the reason I bring this up is again, as we think about what's a being and what's not, this idea that living things somehow generate their own intrinsic purpose and behavior, while machines do the things we told them to do, that doesn't hold anymore. If you're a good engineer, the machine will do what you want it to do, but it's also going to do some other things that you had no idea about. And it's not just the Xenobots and the Anthrobots that are doing that. It goes all the way down to very simple systems. It really doesn't take much. And I'm not talking about unpredictability or complexity. Again, those are easy to get. I'm talking about problem solving and preferences and different goal-directed activities that are literally not in the algorithm. So this kind of thing goes all the way down, and we should be very careful about trying to maintain artificial distinctions between living organisms and machines. And because the thing is, we often run afoul of this issue, which Magritte was reminding us here, that this is not a pipe. This is a painting of a pipe, and people will often say that machines have various limitations. What they're thinking of are the formal models of machines, things like Turing machine formalisms and things like that. Yeah, those things have formal limitations, absolutely. But my claim is that nothing really is the formal models we use to understand them, and minds are not fully defined by our models of them, and machines are more than the materials and the algorithms we think they are following, in exactly the same way that we as cognitive beings are more than the rules of biochemistry that define our lowest components. So for the same reason that we tend not to think that the mechanical laws of chemistry define the story of our mind, those exact same reasons are why the algorithms that we use to model machine behavior are not telling us the whole story. So we need to be very careful with that. Okay, so the last thing I'm going to do in hopefully 4 minutes is the following. So far, everything that we've been talking about is about physical objects, or at least what we thought were physical objects, living things, machines, hybrids, but at least they were all living, at least they were all physical objects. I'm going to take one more step to get even weirder than all of the things I've said before, and try to take you into a new set of potential minds, which exit that set of physical objects. And the thing the conceptual leap we need to understand this is to remember that we ourselves are patterns. Okay, the distinction between physical objects and patterns is really in the eye of the beholder. And I'm just going to tell you a very quick science fiction story. Imagine that there are these creatures that come out of the center of the Earth. They live around the core. They're incredibly dense. They use gamma rays for vision. They work their way up. What do they see on the Earth's surface? Well, they don't see any of us. Because they are so dense, everything that we consider to be physical matter is basically a very thin gas to them. And if they have some tools, so this guy on the left is a scientist, and so what he's got is some tools that can detect patterns in that gas, and he's looking at these patterns, and he says to the others, I've been watching this thin plasma that surrounds the world, and there's some patterns in it. And these patterns kind of look agential. They almost look like they're doing things. They hold together for a little bit, and they kind of move around, and they almost look like they have preferences. And some of them are doing things that suggest they have memories. And the others say to him, well, that's crazy. We're real physical beings. Patterns in a gas can't do anything. Those aren't real beings. And so this is here to remind us that we are also metabolic patterns. We are just sort of temporarily persistent self-organizing patterns like hurricanes and solitons and various other things. And if we are patterns in various excitable media and we are agents, what other patterns might have similar properties? We don't want to be like these guys who don't believe him that the humans, which are basically temporary disturbances in this thin gas, that we don't want to be blind to these to the capabilities of these. So I just want to kind of remind us of what the patterns are. So there are bioelectric patterns here. There are all kinds of these are patterns in a simulated artificial life system called Lenia. So none of these things are actual objects. These are what you're seeing, what your visual system is picking out are basically excitability modes within a medium simulated on a computer. But you can see what's going on here. This is just sort of begging for experiments to see if these things that are holding themselves together for some amount of time, can they learn? Would they show evidence of habituation, associative conditioning, prediction, anticipation? What are they actually capable of? And for biomedicine now, it's very important to start asking, should we be treating the cells and tissues, or are our targets actually the patterns that are propagating through these tissues the way that we propagate through the three-dimensional world? And some people have of course thought about this. Whoops. Such as this idea, and Carl Jung had a bunch of thoughts on this too, that actually we really need to think about patterns, in particular thought patterns, as potential agents. In other words, patterns in any medium, including thought patterns in the cognitive medium of a machine, of an AI or of a brain, whatever. Those patterns might actually have a life of their own, and we now have the tools to study this, and really to dissolve this boundary between thoughts and thinkers. And this is, again, William James said this in his book, that the thoughts can be thinkers because patterns can spawn off other patterns. We are patterns, but the patterns of thought in our brains are also patterns. And so you can sort of imagine this kind of continuum where you can have patterns that come and go, these sort of fleeting thoughts that come and disappear, but some of them hang around for longer. And so you could have these intrusive / repetitive thoughts, and some of them really hang around. Oh, and by the way, these do some niche construction because having those kinds of thoughts makes it easier to keep having more of those thoughts. They actually change those kinds of thoughts change your brain. And then, of course, you might have like a much richer kind of thing, like a personality alter in dissociative identity disorder. And then, of course, there are even bigger ones that are full human minds. Who knows what's beyond this? And the very last things. Okay, so what I'm trying to tell you here is that the potential set of the innumerable set of agents, it's not even just the physical things that we can see. It might be patterns within medium that we've been completely, basically blind to, and treating them as these mechanical propagations through the medium. There is this spectrum between whatever we are doing as patterns within this cognitive medium and other kinds of patterns. And so the last thing I'm going to show you is just to say that, okay, all of these patterns, but at least they're set by evolution, right? Evolution has determined through past history, genetics, whatever, determines what these patterns are. I just want to remind ourselves that in mathematics, we have very rich patterns that are not determined by either the laws of physics nor any kind of past history. So this is what's called a Halley plot of a complex number formula like this. This little tiny seed is hiding all of this. And if you start changing these numbers, you can get these amazing videos, and you can get things like this. The idea, but basically, is this: there are patterns that come from mathematics that are not explainable or changeable by anything you do in the physical world. Okay, so to explain why this is the way it is, there is no story of physics, there is no story of history like evolution. This comes from an entirely different world. So as we think about what determines the form and function of things like Xenobots, of things like cyborgs and hybrids, and these crazy, diverse systems that I've been showing you, what determines their psychology? Where does their psychology come from? Well, we need to understand that there's an input into all of that is not genetics, it is not chemistry, it is not physics, it is wherever it is that mathematics comes from, and that's a very deep and profound question. You know, mathematicians think that there is this, at least many of them think that there's this abstract Platonic space that contains laws and facts that are not determined by physics, basically. And I think that's right. And I think that space also contains patterns that we can recognize as kinds of minds. They're patterns of not only form, but of behavioral propensities. And so when we make things, be they cellular structures, embryos, AIs, whatever, you know, humans, whatever it is that we make, these physical objects are basically pointers or interfaces for these patterns. And some of these patterns are simple things like facts about triangles and Fibonacci series and so on. And some of them are very rich, complex patterns that we recognize as kinds of minds. And so this is what I think that the opportunity here is before us, is to start to characterize, use some of these things as periscopes into that space and start to characterize what is exactly the option space. And so this is actually what I think you know, the real Garden of Eden is going to look like. It's going to be very weird and very divergent. And we now have the beginnings of some tools to expand our mind blindness into the ability to see way beyond the brainy things or the biologicals or the physical objects even. And there's one other step that I didn't have time to talk about that, you know, we'll deal with that later. And so I'll just stop here. I'm already over time, so I'll stop here just to point out that we are going to be living in a world where all of your familiar biologicals, what Darwin called the endless forms most beautiful, all of them are a tiny little corner of this option space. Okay, every combination of evolved material, engineered material, and AI is some kind of possible being with the cyborgs and hybrids and all these different chimeras and so on, because all of them, including the simple machines, are pulling patterns from this latent space of form and function that we are very bad currently at predicting. And so I will here. I'm going to stop here by just thanking all the folks that did some of the biology that I showed you. So these are all my amazing postdocs and PhD students and others in the lab. We have lots of incredible collaborators. Do have to do a disclosure. There are 3 companies that have spun out of our lab and are funding some of this work. And as always, I thank the model systems for teaching us about all these things. So I'll stop here and thank you.
Thank you so much. That was absolutely incredible. I'm just like trying to process, but amazing. Thank you for sharing that with us. I know it's going to be a huge hit, especially once we get to post it with our global audience. But do you have time for like a couple of questions, or you need to go?
Yes, I do. Let me just, I'm just going to email real quick my 4 o'clock that I'm going to be late. All right, yeah, go for it. Just a couple of minutes. Yep.
Okay, so one person asked, is there somewhere we can see these slides? Yes, actually, that was one of my questions. Is it possible to share these?
Yes, I can do that. Send me an email later and I'll make them available.
Perfect. I'll put it on our website for everyone so they can see it if you haven't. And then somebody else asked, can you speak to the nature of self-organization and how this is seen in your research?
Yeah, basically, there's two ways that people think about self-organization. Conventionally, when, for example, when a gene regulatory network is self-organized, meaning that there's a random set of nodes, they're connected in specific ways, and then we find out that, hey, this thing's capable of associative conditioning. You say, hey, why did that happen? Why did it self-organize in a way that enables it to do this kind of learning? Typically what people say, because they want to keep to a physicalist worldview and a pretty sparse ontology, they will say, well, that's just a fact that holds in this physical world, just a fact that holds. And so it's emergent. Oh, the other thing they'll say is that it's emergent. And so if you actually dig into that, I find that what that's basically doing is trying to it basically makes it sound like you've said something that means something, but really when people say these things are emergent, what you're really saying is it's surprising. What you're really saying is that my formal model of this did not see that coming. So it's emergent. And as soon as if you did have a formal model that would made it predictable, then you would say, well, it's not emergent, it's obvious. You know, those are the 2 options. It's either obvious or it's emergent. I don't like the emergence part because I don't like this sort of Mysterian view that there's this random grab bag of things that happen to hold in our world, and then we're just going to sort of write them down in our big book of emergence, and when we come across them, and that'll be that. I don't like it. But now, of course, I can't prove this, but it's a metaphysical assumption that I make and that a lot of mathematicians make. which is that no, these things are part of an ordered space. They're part of an ordered space which we can systematically investigate. And exactly the way that mathematicians are making the map of mathematics, but I think it actually goes way further than mathematics, and we can systematically explore it so that when we see an example of self-organization, we can actually eventually we'll be able to say, here are the features of the materials and the algorithm you've put together that allow us to index into the space of these Platonic patterns, which is why you're getting what you're getting, right? So there's a point that where normal frog embryos bring down a bunch of frog related patterns, but in that neighborhood, there's a different thing you can index into, which is the Xenobot way of life, and who knows what else is there. So I think as long as we stay away from this Mysterian view of emergence as just surprising things for us to note, we can start systematically figuring out what are you actually pulling down when you have an example of self-organization.
Perfect, thank you. Are you okay for 2 more?
Let's do one more.
Okay, so the next one is, if we think of exocrine signaling or plant VOCs as Bluetooth, maybe we can channel that to communicate with antrobots like the robots in the arena analogy.
Sure. Yes, that certainly sounds possible. We don't know yet what they can sense. We have a few, we have some data, but yeah, for sure, all of those things, chemical, electrical, haptic, optical, all of those interfaces need to be explored. We have no idea what these things are actually paying attention to.
Perfect. Thank you. I know you got to go. Thank you so much for staying over time. It was an absolutely mind-blowing talk. I can't wait to share it. And thanks again for being here. I really appreciate it.
Thank you so much. Yeah, thank you both. I appreciate that very much. Anybody that has questions that I didn't get to, I'm sorry. Send me an email and I'll answer everything.
Perfect. I'll show your information. Thank you so much.
Thank you.
Thank
Thank you,
you.
Dr. Levin.
Bye-bye.
Thanks a lot. Good to see you.